Hello everyone. We will now be discussing about the gram stain and its procedure. The gram stain was developed by Christian Gram in 1884 and modified by Hucker in 1921. This test differentiates the bacteria into gram-positive and gram-negative bacteria, which helps in the classification and differentiation of microorganisms. The gram stain separates bacteria into two groups. One, it's a gram-positive microorganisms that retain the primary dye crystal violet, and number two is a gram-negative microorganisms that take the color of the counterstain safranin. Gram-positive organisms contain a highly cross-linked layer of peptidoglycan that retains the primary dye crystal violet. Following the application of the modern iodine, the iodine and crystal violet form a complex within the peptidoglycan when decolorizer is applied to the cells. The CVI complex remains within the cell, making it appear dark purple to blue. The gram-negative organisms do not contain a thick cross-linked layer of peptidoglycan. The peptidoglycan is loosely distributed between the inner cell and the outer cell membranes. Following the application of the crystal violet and iodine, the CVI complexes are not trapped within the peptidoglycan. Application of the acid alcohol decolorizer dehydrates the outer cellular membrane, leaving holes in the membrane and effectively washing or removing the CVI complex from the cells. The cells appear colorless. To make the colorless cells visible, a secondary stain, safranin, is applied, leaving the gram-negative cells pink. So now we are going to start with the procedure of gram staining. At first, we need to prepare and fix the specimen to the microscope slide before staining. So prior to that, we need to properly sterilize the inoculation loop. The inoculation loop has to be red heated in the Bunsen burner that we are using for the sterilization process. Even the slide which we are using has to be flamed before the smear is made to it. We have a 24 to 48 hours temperature in the petri plate, and the plate is open near to the Bunsen burner so that there is no contamination, and a loop full of the culture is taken on the inoculation loop and the smear is made. We are preparing the smear on the sterile glass slide. The smear has to be proper so that the staining is properly done and we are able to visualize the organisms. The inoculation loop is again sterilized and then kept aside. And now we need to heat fix the smear that we have prepared. The reason why we need to heat fix the bacterial sample or the smear is before staining is because heat fixing attaches the smear to the microscopic slide, and it also allows the sample to more readily take up the stain. So in order to heat fix a bacterial smear, it is necessary to first let the bacterial sample air dry properly. Then we allow the slide to be passed through the flame. So here we have the smear prepared. We take the reagents. This is the grams crystal violet. This is grams iodine. This is grams decolorizer. And finally we have the safranin. So first of all, we will cover the smear with crystal violet, which is the primary stain. The entire smear is covered with the crystal violet stain, and then it is allowed to stay for 30 seconds. We'll have to keep a proper check on the time. As the 30 seconds gets over, we need to gently rinse off the stain with water. Wash it away with water, keeping in mind that the smear is not washed off. Then we cover the smear with grams iodine, which is the mordant. The grams iodine is allowed to stay on the smear for 1 minute. We'll keep a check on the time again, and after that we pour off the excess grams iodine. Then we run the acid alcohol decolorizer or the grams decolorizer over the smear until the solution appears clear, and gently rinse with water. After gently rinsing with water, we will cover the smear again with safranin. Safranin is the and then we will let the safranin stay over the smear for 30 seconds. Once 30 seconds is over, we will gently rinse the stain with water. After that, we need to blot dry the glass slide blotting paper. We need to properly allow it to dry, keeping in mind that the smear is not washed away. The sides or the corners of the slides are also dried. The water is completely blotted off. And then we let it stay for some time, allowing it to once the slide was air dried, we observed the slide under the microscope at 100x. On observation, we found the presence of purple color colonies which are rod shaped, which indicates that the bacterial sample that was stained is a gram-positive bacilli. So this is how we perform the gram stain.